Okay, let me start with a quotation from Siddhartha Mukherjee, who said that medicine asks you to make perfect decision with imperfect information. So this is about related to cancer diagnosis, okay, where we'll be talking about how to make this imperfect scenario a perfect one. But before that, I would like to thank Dr. Rao, Dr. V.P. Rao, and Global Cancer Summit for the opportunity to be here. Okay, let me start with 1953, which is about 60 years ago. There were two landmark papers. One was from Nordling, and the other one was from Slaughter. What did Nordling say is that, he proposed this multi-stage mutation theory that is within the stem cell, you have a mutation, and then when it grows, more and more mutations get accumulated, and finally it results into what we call cancer. Now, at the same time, there was another group of people led by Slaughter who talked about something which we called field cancerization. What is field cancerization? He examined 783 patients, and he found that each and every patient, they had the histological properties of the tumor cells in non-tumor cells. So his discovery was that the properties which you see in the tumor cell are also seen in non-tumor cell, which are close to the tumor. And here, these are the from his original paper. And here is what he said, 783, each and every of them showed that property. And then in last 60 years, a lot of other work in molecular biology and experimental biology has proven over and over again that cancerization or field cancerization is a property which is not only seen in the tumor, but it is also seen outside of the tumor. And here are some of the publications for last 60 years. So why we are here? What's my interest? Now, the point is that what Slaughter has proven, and the last 60 years of research has proven, what we call is the cancer field. Now, if you look at the magnetic field of the world, you can have a compass. And if you have a compass, then you will be able to sense that from a distance. And it has already been proven that you can see the signature of cancer in non-tumor cells. So the question we asked, is it possible to see that signature in the blood? Now, what is the advantage? The advantage is that if we can sense that in the blood, then we don't need to have the tissue biopsy, number one. Number two is that if I'm able to get that from the blood without the morphologic lesion, I'll be able to see the cancer, which might lead to the early detection of cancer, which all till now people believe a myth. So having said that, and this what we call is the blood biopsy. And I'll take you through some of the research work and the results. So some time ago, we had a publication on exome data. And this is all data-driven mathematics and computing or computer science-driven biology. So where we took the exome data, and from there, we did identify the mutations and some of the novel one and other stuff. So this is reported here. Now, I'm going to show you some of the exciting findings, and this is from a breast cancer cohort, and the cohort size is here, 31 patients, and you can see very clearly here that that the healthy and the cancer patients, they are clustering very clearly. Next is ESCC cohort, which is of 38 samples or patients. Now here, what you can see is that as Slaughter has said, that you can see the signature of cancer or your metastatic cells into the normal tumor. And here is the normal patient or normal individuals and the ESCC patients, and you can see here that they are pretty intermixed. So what the experimental finding of 60 years has proven in biology, we are seeing that through mathematics and computer and through analytics. So the same one, you can see some details. And the statistics when we did, we found that 99% of the genes, top 100 genes, are common between the ESCC tumor and non-tumor cells. Here are some of the p-values, and p-value goes up to e to the power minus 102. That is the statistical significance, and that's how you can quantify the level and importance of this. Then we took breast cancer cohort, and this breast cancer cohort has total 84 samples. Out of that, 20 are healthy, independent healthy individuals, and rest are all breast cancer patients. And if you see here, once again, here if you see that tumor, non-tumor cells are combined, or they are, I would say that it is difficult to discriminate between them. And for this cohort, we also had blood, okay? So here is the, what I would say, the molecular signature of blood, molecular signature of adjacent tissue, along with the tumor tissue, the matching tumor tissue from the same patients, okay? They are clustering together. And you can see that how beautifully and mathematically they can be identified. The advantage of mathematically and computationally doing it means that we'll be able to do it and repeat it with a lesser cost. As once those are all tested in the clinical trial, then it can be taken to the clinics where the cost will be much more affordable than what it is today. And the statistics here is that 89% of the top 100 genes between the tumor and non-tumor cell, including the blood, are common. And some of the pathways which are implicated are here, that is cell addition, cell selection, so on and so forth. Now, the question is how to take it to the clinic. So we have proven, or we have proofs for last 60 years that there is a cancer field. So our objective is to find out that, like Compass, can we have a device which will be able to sense that, and that also mathematically and statistically significant. So having said that, what we did was that in this, then we took the bladder cancer data, which is of 52 samples. Here we have taken the blood, or the tumor and the matching blood of the same patient. So the cohort size is 52. And there, once again, you can see that how this beautifully clusters, and we are able to now mathematically say that this is what is the line between cancer cell, non-cancer cell, and the healthy tissues. And there, once again, if you look at the statistics and numbers, 97% of the top 100 genes are common between the non-tumor and tumor cell. And here, non-tumor is the blood. So having said that, the question is that, okay, mathematics is fine. So mathematically, statistically, and otherwise, we have proven that whatever has been seen for last 60 years, that is the cancer field, is also detectable through devices, through scientific techniques, and can be done without the wet lab. However, this wet lab is practically NGS, or the next generation sequencing data coming from those tumor, non-tumor, or blood cells. So then we did the function analysis, and from the function analysis, we are finding that our finding, not only mathematically, but biologically, is exactly matching of last 60 years of research. And then the same is true when we took the blood and did the functional analysis on that, on the top genes and the significant genes. They are matching. And here you can see also some other analytic analysis where we have done something which is graph analysis, which is primarily you do in social networks. In a social network, I'm sure many of you have Facebook or some other account. So when you go there, it suggests that, do you know this person? He could be your friend. So would you like to connect? So there are mathematical properties between all those different kind of, what I would say, the graph theoretic point of view. So here it is not visible, but what we also proven that along with the pathway and other type of analysis, there are very high level of similarity between the tumor tissue and the blood, the matching blood of the cancer patient. So then what we did is that we identified 24 genes which are primarily related to oncogenesis, and then from there we created a panel for the bladder cancer. And you can see the result of those 24 genes which has been put into the panel, and then look at the result. Here, the cancer patient genes, their expression. And now, and just mind you, here I'm not talking about expression as my lab. What we are doing is that we have designed new algorithms, what we call is DNA profusion. What does DNA profusion does, as the English word signifies, is that how the DNA of an individual is spread and mathematically measure it, so quantification. So our algorithm, what we call is DNA profusion, and then what we do is that take the blood sample of the cancer patient and then put it in the NGS exome analysis, and then quantify it from what generally in molecular biology is called CNV, copy number variation, as we already have panels where we are doing mutation test, where we are confirming, yes, somebody has this type of cancer or likely to have that. And when I'm talking about likely to have, I'm only talking about germline mutations. However, with this blood biopsy, we call this technology and the science. More than technology, it's a science, as I have shown you the different kind of mathematics and numbers and quantification. The paper is now getting ready, and hopefully it will be published very soon, that we are able to detect the signature of cancer in the blood. And that is why we call it blood biopsy. Before I finish, let me just tell you, this is not liquid biopsy. This is not liquid biopsy where you take the blood and then try to look for circulating tumor cell or other type of cells or cell-free DNA, and then you try to find out the mutation. It is not that. It is peripheral blood. Once again, from that peripheral blood, we identify the signature, and mathematically, we are able to identify. And right now, we are recruiting patients. Hopefully, within a short period of time, we'll be able to prove that it works on a larger population. Thank you very much for your time and patience. I'll be happy to take questions.
Thank you, Dr. Ashok, for a wonderful presentation on blood biopsy and differentiating it from liquid biopsy and telling how you can utilize it in analyzing the various circulating tumor cells.
Oh, before I just finish.
One question.
Before I just finish. So this is the pipeline. Okay, what you do is that you take healthy or early stage cancer, then you draw the peripheral blood once again, and then you do a targeted exome sequencing using NGS technology, and then use this model. We call it the mechanistic model of cancer field, where mechanistic means that mathematical algorithms and computational and all those things. And then after that, if you see the p-values are not significant, you know that the chances of cancer is very less. And if the statistics is very, very significant, we can say that he is likely to be a patient. So use a blood biopsy of selected cancer-filled biomarker genes in blood and decide clinically whether the individual has cancer or not. If not, whether there is any possibility or probability of this individual to have cancer at a particular date, looking at the probability and mathematical values. Thank you.